This is the second recording for the Chapter 13 notes, and we're starting with slide number 22. Still talking about the life cycles of organisms that reproduce sexually. We already, actually, slide 22, we've already covered that information. So the life cycle in an animal is compared to plant and fungi, and we are going to skip that. I think I mentioned to you I'm going to concentrate more on the human life cycle. In Bio 112, you will learn that the life cycles are slightly different in other organisms, but we're going to just concentrate on humans, really. So now we're going to move on to section 13.3 and learn about meiosis in detail. Okay, so we already did mitosis. We know the main stages are interphase, prophase, metaphase, anaphase, and telophase. And thank goodness in meiosis we have the same phases with some slight differences in some of those phases. Okay, so one thing about meiosis that is similar, that's the same as mitosis, is that like mitosis, meiosis begins with interphase. So there is an interphase at the beginning of meiosis, and during interphase we know that chromosomes are replicated, they're duplicated. So you start with 46 single-stranded chromosomes, and after interphase, after the chromosomes are replicated, we have 46 chromosomes that each have two sister chromatids that are attached at a centromere. So that's going to be interphase, and that will come before meiosis 1. Now we get to where meiosis is different from mitosis. Meiosis has two cell divisions. The first one is called meiosis 1, and the second division is called meiosis 2. And in meiosis, after the second cell division, you end up with 4 daughter cells, and each of those daughter cells has only half the chromosomes of the parent. So they are haploid rather than diploid. Okay, so this is an overview, which we actually already looked at this same exact slide. It was part of the one where we looked at the mom and the dad's chromosomes in the last lecture. But anyway, you start with a cell, a parent cell, and this could be in a cell that's in the testes of the father or the ovary of the mother, but it's going to have the diploid number of chromosomes, and after interphase, they will be duplicated chromosomes. So each pair of chromosomes will contain 2 sister chromatids. Okay, so in meiosis 1, each pair of chromosomes will separate, and so you end up with 2 daughter cells that are haploid, and those 2 daughter cells will divide again, and this is meiosis 2, and this time it's the sister chromatids that will separate. So you will end up with a total of 4 daughter cells. And if this happened in a man, they will be sperm. If it happened in a female, they will become eggs. So the phases of meiosis 1 will begin with meiosis 1. The phases are called prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. And sometimes cytokinesis doesn't occur all the way in meiosis 1. The cells don't divide all the way, but they do at the end of meiosis 2. But that's not really a big deal. What I really want you to concentrate on are the events that occur in prophase 1, metaphase 1, anaphase 1, telophase 1, and how they are different from mitosis. Now, we already know in mitosis that during prophase, the chromosomes condense. In interphase, the DNA was in a form called chromatin, and it was very spread out and diffuse throughout the cell. So when you looked at the nucleus under the microscope, it was like a solid color. You couldn't see the individual spaghetti strands that we call chromosomes. Okay, but during prophase, the chromatin condenses and forms chromosomes. So that also still happens. Remember, the nuclear envelope also disappears in prophase. That still happens in meiosis. So another thing that happens, though, that's different from mitosis, and you need to know this because it's a very, very important part of prophase 1, and it has to do with genetic variety, how we get genetic variety. So in prophase 1, a process called synapsis occurs, and that is when each pair, each chromosome that forms a homologous pair, they find each other and pair up. So each chromosome pairs with its homolog, and then something called crossing over occurs. And crossing over is when some of the DNA is exchanged between the chromosome pairs. So you get even more variety than you would otherwise. And we'll look at a picture of that so you can understand it. But just remember, for prophase 1, crossing over occurs, and that is different from prophase of mitosis. We don't have crossing over. We don't even have the homologous pairs don't match up in mitosis. All right, in metaphase 1, I can't highlight that. Yes, I can. Okay, it's kind of this. All right, in metaphase 1, the homologous pairs line up at the metaphase plate. So instead of the chromosomes lining up individually, they line up in pairs. In anaphase 1, the homologous chromosome pairs separate. Oops. And I don't know why that must have to do with how something I did. I don't know, but the homologous pairs separate. If you remember from mitosis, it was sister chromatids that separated. But in meiosis 1, it's the homologous pairs that separate in anaphase. And then in telophase 1, in cytokinesis, each chromosome is still going to have two sister chromatids. The sister chromatids haven't separated, but you only have one chromosome from each pair in the daughter cells that form. So the daughter cells are now haploid at the end of telophase. Okay, and there'll be two. There'll be two haploid daughter cells. And then we will have, this is a picture. So then we will have those two daughter cells will then divide in meiosis 2. But let's look at the picture and talk about meiosis 1, okay? The overall goal of meiosis 1, the result of meiosis 1 is that meiosis 1 separates the homologous chromosome pairs, okay? That's what happens in meiosis 1. So you can see prophase 1 that the homologous pairs are finding each other, they're pairing up. We call that synapsis. And you can also see, even if you may not understand it that well, that some of those sister chromatids, from one from the mother's chromosome and one from the father's, will cross over and change places. So it gives you even more genetic variety than you would get otherwise. So we have crossing over that occurs in prophase 1. The homologous pairs match up and crossing over occurs. And then in metaphase 1, the homologous pairs line up at the metaphase plate, whereas in metaphase of mitosis, it would have been 6 chromosomes lined up in a row. Now it's going to be 3 pairs of chromosomes lined up. And then the homologous chromosomes separate in anaphase, and in telophase, you end up with 2 daughter cells that have the haploid number of chromosomes. So now we're going to go over meiosis 2, and it's very similar to mitosis. So meiosis 1 is the part that has, differences from mitosis, but really meiosis 2 is just like mitosis. So meiosis 2 has 4 phases: prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis, and it's going to be just like what you learned in mitosis. In prophase 2, the spindle is going to form, the spindle fibers or microtubules are going to attach to the chromosomes, and they're going to begin moving those chromosomes toward the metaphase plate, preparing for metaphase. So in prophase 2, you just basically have the attachment of the chromosomes to the spindle fibers. In metaphase 2, the chromosomes are going to line up at the metaphase plate, individually. There's no crossing over. And then in anaphase 2, make sure you get the wording here. In anaphase 2, chromosomes separate, but we don't say homologous chromosome pairs separate. We say sister chromatids separate. Sister chromatids separate. In anaphase 2, just like in mitosis. And then in telophase 2, you're going to have the chromosomes have arrived at opposite poles, and the nuclei the nuclear envelopes are going to reform around them. Cytokinesis will occur, and you will end up with 4 haploid daughter cells at the end of meiosis 2. 4 daughter cells, each with a haploid set of chromosomes. And this is what meiosis 2 looks like. Okay, you start out with your prophase. You have the haploid number of chromosomes, but they are duplicated. They each have sister chromatids joined at a centromere. In metaphase, they line up individually at the metaphase plate. In anaphase 2, the sister chromatids separate, and then in telophase 2, you're going to end up with, I'll show you the result after, well, it doesn't. Well, after telophase 2 and cytokinesis, you will end up with the haploid number of chromosomes in each of the 4 daughter cells, and they will be single-stranded chromosomes because they are just one sister chromatid, from the duplicated chromosome that was in metaphase. Okay, so they just consist of single chromatids, not sister chromatids. Now, this is a good animation to watch on meiosis. I would absolutely watch it. It's slide 47. And then we're going to talk a little bit more about crossing over. The process where during prophase 1, where the homologous pairs find each other is called synapsis. And the process of crossing over occurs during synapsis. So this is a picture of what crossing over looks like. Basically you have, I hate this picture. Oh my gosh. Okay, let me just show you using this picture. Because they're not showing you both of the sister chromatids, and you can't really understand, I don't think. Okay, so I just want to look at this cell because it has the homologous pairs have paired up, okay? And you can actually see crossing over occurring here. So when the homologous pairs pair up, you can really even see it better right here. So we're going to look, this is actually showing you prophase 1. All right, so we'll just say that the blue chromosome is from the father originally and the red is from the mother originally. Let's just say that, okay? That's just, I don't know, but we're just going to say it. All right, so the blue chromosome from the father has 2 sister chromatids. Then the red chromosome from the mother has 2 sister chromatids. They are each duplicated chromosomes, right? Okay, the chromosomes that cross over are the 2 that are closest to one another from the father and the mother. They're called, now what we call them is non-sister chromatids. They're not sister chromatids because they aren't from the same chromosome. There's one from the blue and one from the red, and you can see that what happens is they cross over and literally exchange pieces. So where they crossed over, this red chromosome is now going to have a blue piece on the end, and this blue chromosome is going to have a red piece on the end. And that is going to increase the genetic variety. So that process where the homologous chromosomes pair up, we call that synapsis. And then when the non-sister chromatids exchange segments, we call that crossing over. All right, so these are the events that are unique to meiosis, and they all occur in meiosis 1. During prophase 1, we have synapsis and crossing over. And then in metaphase 1, we have the alignment of the homologous pairs at the metaphase plate rather than the chromosomes lining up individually at the metaphase plate. If you can visualize this in your mind, if this were occurring in a human cell, during metaphase 1, you would have 23 pairs of chromosomes lined up at the middle. But if it were mitosis, you would have 46 individual chromosomes lined up at the middle. And then anaphase 1 is when the homologous pairs separate rather than sister chromatids separating. And here's a comparison of mitosis and meiosis. The main thing is the end result is in mitosis, the end result is 2 daughter cells that are each clones of the parent. So they have the diploid number of chromosomes. In meiosis, you have 4 daughter cells that are haploid. There is a section here on how genetic variation contributes to evolution, but I just want to focus on the 3 ways that we get genetic variation, the 3 mechanisms that contribute to genetic variation. And 2 of them occur during meiosis. We already talked about crossing over, but there's also the independent assortment of chromosomes, and that happens in metaphase. And then there's random fertilization during fertilization. Okay, so independent assortment means that during metaphase 1, you know the pairs line up, right? So if we go back to this picture and look at metaphase 1, you can see that the top homologous pair, there is the dad's chromosome lined up on the left and the mom's on the right. And then the last 2 pairs, the mom's chromosome lined up on the left. Okay, well, in the homologous pairs, the father's chromosome and the mother's chromosome randomly line up. So sometimes the father's chromosome is on the left and sometimes it's on the right, and that is literally a random thing. And it does result in different combinations in the daughter cells that are produced. So that is called independent assortment, is the way that the homologous pairs line up during metaphase 1. That's independent assortment, okay? It's the lining up of the homologous chromosomes during metaphase 1 of meiosis 1. That contributes to genetic variety because you end up with different combinations of genes in the offspring, in the sperm and the egg, so it would be in the offspring. And then crossing over contributes to genetic variety, and then the actual sperm and the actual egg that unite during fertilization contributes to genetic variety. So let's talk about independent assortment. All right, so if we have only 2 sets of chromosomes, we have 4 possible combinations of those chromosomes at the end of meiosis. In combination 1, let's just say these are all going to be sperm, okay? So you could have 2 blue chromosomes in 2 of the sperm. That's one combination that could be produced. You could have 2 red chromosomes, or you could have a blue and a red, a long blue and a short red, or you can have a long red and a short blue. So there's 4 possible combinations if you start out with 2 sets of chromosomes. And one of your questions on your practice questions for chapter 13 asks you how many combinations could you get just from independent assortment if you had 3 chromosome pairs. And in that case, if you had 3 chromosome pairs, you could get 8. You could get 8 different combinations. And there's a way to figure that out, but I'm just telling you the answer right now. So crossing over contributes to genetic variation. Oh, this is a great picture of crossing over. You see how after crossing over, you end up with one of those sister chromatids is a combination of pieces from the blue and the red. So that gives you even more variety, more genetic variety in the sperm and the eggs that are produced. And then the last way that we get genetic variety is just random fertilization, because any of the sperm that are produced can combine with any of the eggs that are produced. And that's just, it's random which sperm fertilizes which egg, okay? All right, and I'm going to stop here. So that's the recording for chapter 13.